Se abre la sesión. Tiene la palabra el señor Philippe Gillet, coordinador del proyecto Human Brain Project.
Your Majesty, Secretary General, colleagues, students, guests. It is a great pleasure and an honor to have you with us here in Madrid for this third Human Brain Project Summit. It is a key event. We have around us people that have undertaken a fabulous scientific challenge, understanding the way the human brain functions using data, simulation, and models. A unique journey in modern science with experienced scientists and engineers, but also with a new generation, the one that will carry the torch in the next decades. Being a scientist is a fabulous job, a job in which we address the fundamental questions concerning humankind. Since the very beginning, science provides pieces of answers to numerous questions that are all more or less included or summarized in the famous Shakespeare's questions. Where do we come from? Who are we? Where are we going? Three short examples to illustrate my purpose. Where do we come from? When astrophysicists recently looked at the data collected by the European satellite Planck, they were just observing the remaining light from the initial explosion that created the universe in which we live in. When they landed a few months ago a spacecraft on the Chury comet, it was to look for molecules that might have participated to the emergence of life on Earth. Where are we going? The central question for the next generation. A question that has to be tackled jointly by scientists and engineers in close connections with citizens and politicians. Preserving the planet, making reasonable use of the planet's resources, nurturing a growing population, providing health care for an aging population are among the most central issues we all have to face together. Who are we? Our brain defines us. Understanding the human brain is one of the greatest challenges facing 21st century science. If we can rise to the challenge, we can gain profound insights into what makes us human, develop new treatments for brain diseases, and build revolutionary new computing technologies. Today, for the first time, modern information and communication technologies have brought these goals within sight. These examples show common features. They rely on what we call big science, the science that needs big human and infrastructure investments. Through this project, neuroscientists are taking the same steps other successful branches of science have taken before. What is the Human Brain Project? One of the biggest programs ever financed by Europe, a European Commission Future and Emerging Technologies flagship. There are only two of them, the other one being graphene. A very large-scale European collaborative research initiative, a 10-year research roadmap with a budget of €1 billion shared between the European Commission and the partner countries. More than 500 scientists from 112 institutions representing 24 countries, most of them being European, including, of course, Spain. Your country, Your Majesty, has played a key role in this project. Javier de Felipe from Madrid and other pioneers joined the Blue Brain Initiative early on, the seed of HBP that was launched by Professor Henry Markram in Switzerland 10 years ago. This summit will be an opportunity to see the amazing work carried out in the past year since the previous summit held in 2014 in Germany. Dozens of papers, new computer programs simulating brain activity, amazing discoveries made by the young talents that are here today. It is important that European countries support such challenges despite the difficult economic times. Other countries like China, Japan, or the USA, following HBP, finance similar initiatives. Europe must not miss the discovery train of the brain. A lot of innovation is expected from the HBP in the coming decades. The journey will be long, but it is so exciting and promising, and we all rely on the continuous support of Spain. Thank you very much.
Tiene la palabra doña Sonia López, presidenta del Ilustre Colegio Oficial de Médicos de Madrid.
Her Majesty, the Queen of Spain, Sofia, His Excellency, the Secretary General of Health, Dr. Javier de Felipe, ladies and gentlemen, good morning and welcome to all this historical site of Illustrious Medical Association of Madrid, where the Human Brain Project Summit is taking today. My name is Sonia López Arribas. My brand is Psychiatry, and I am the president of the Medical Association of Madrid. First of all, I would like to thank Her Majesty the Queen, Sofia, for honoring us with the presence in this event. Her Majesty is very well renowned in Spain for her tireless impulse to culture and welfare initiation for many years now. And it's certainly a great honor for us to come with her presence. Thank you very much. Your Majesty, thanks a lot for coming to this historical building and for staying with us today. Now, I would like to highlight that we are now sitting on the historical site of medicine in Spain. We call Professional Association College, which is the Spanish word for college, because colegios is for us the name. This association was born in Spain as a teaching institution under the protection of the monarchy. The early Spanish surgeon college date back to the mid-18th century. The first surgeon college in Madrid was founded in 1708, not far from here. The building in which we are now was built in 1834 and was the new site of the Royal College of Surgeons of San Carlos. It has been used as a College of Medicine until as late as 1965. Generations of physicians have studied in these classrooms, and some of the most important doctors in Spain were our teachers. The most famous of them was the undoubtedly Dr. Santiago Ramón y Cajal. He is documented to have been a member of our Medical Association of Madrid for 34 years, since very late in the 19th century, nearly until his death at the age of 82. Dr. Ramón y Cajal was not only the neuroscience that we all know today, but he was a pathologist and histologist as well, and also a medical captain in the Spanish army for some time. He was also a gifted painter, what allowed him to produce hundreds of excellent drawings of his discovery, like the one we have, he painted when he was 12, and it's a little lady, and it's just beautiful, the picture. He was vested in honorary doctorate in medicine from the Cambridge University and Oxford University, among some other distinctions, and was finally awarded the Nobel Prize in Medicine in 1906, shared with the Italian Camillo Golgi, in recognition of the work of the structures of the nervous system. But as well as I have said, this building was now at the late 19th century the site of Medicine College of Madrid, and Dr. Santiago Ramón y Cajal was able to teach medicine from this very place. Most of the halls at the chambers of the building are still called classrooms today. As a matter of fact, you may visit the hall, and that now is called the Ramonica Hall classroom, where he taught his students more than a century ago. In raw, the same appearance it saw at the time he was teaching. It is still used as a classroom today for our courses of medical, medicine and continuous formation period for the new doctors. Because of all that, it's a deep satisfaction for me that you have chosen the historical building of Medical Association of Medicine for this Human Brain Project Summit, returning these amphitheaters and hall to the ancient vocation and which they were designed, built and decoration, and from which Dr. Santiago Ramón y Cajal showed his discoveries to his students for sharing the medical knowledge. I hope that you make yourself at home in these chambers and that these days you work up and finish with your expectation about the Congress. Thank you very much for your time, and I wish you a pleasant time here. Thanks a lot.
Tiene la palabra don Rubén Moreno, secretario general de Sanidad y Consumo.
Your Majesty, good morning to everyone. Being here among all of you, members of this extraordinary collaborative effort, which is the Human Brain Project, it reminds me of another project I was part of, which was the Human Genome Project. I was working at that time in the US before any automatic sequencing machine was available, and obviously looked at that time an extraordinary task. Altogether, that project, it was more certain probably than your own one. After all, we have to sequence 3 billion base pairs, and to understand them, which is something that still we are working on. But what you are trying to do, among many other things, is to somehow, for the lay people, to reconstruct informatically, digitally, human brain. Let me tell you something about the human brain, because at that time I was working at the National Institute of Neurological Diseases and Stroke. Being so difficult to sequence the human genome at that time, we decided to sequence small pieces of that, which is the genes. But those genes were totally unknown at that time. We decided to use our own body to help us to find those genes. How we did that? We pick up some samples of human brains. After all, we were at the Neurological Disease Institute at NIH. Some brains that we brought from a brain bank in Boston. And we tried to copy what those cells, before the person died, was doing to help the brain to work as the brain is working. So we copied what we call messenger RNA, and we made cDNAs, and then we sequenced that. But this was, after all, what this brain was using. Several lessons that we have probably to remember from that experience. First, and you know that better than me, there is not such a thing like a human brain as itself. We tried to do the cDNAs from the embryonic stage to the end of the life of this person. And let me tell you, the messages were completely different through all of these developments. So there is not a brain, as you perfectly know. There are many stages of this brain development, one thing. The second important lesson that we have experienced with this brain project as well, the criticism and the criticism in this kind of projects cannot come from anyone else but from our own people in the field, neuroscientists and so on. The criticism that we had at that time, very tough, really very, very tough. The worst was Jim Watson, Nobel laureate, who discovered the DNA helix, and having him as an enemy, let's say that, it was a difficult task to go over. So don't worry about the criticisms in this project. That's the main thing. You will hear that 1,000 million euros is a lot of money. And other people, especially from my specialty, I'm at the ministry now, but at that time I was working in molecular biology and I'm pathologist as well. The people say, well, if these people are using such amount of money, it's because I'm not going to receive my own part. So this is how these things are really working. So don't worry about that, just keep working. And the second thing is that, again, I mean, difficult is to sequence the DNA and understand the DNA. Much more difficult is going to build a human brain in the way that we are trying to. But at the end, this is going to help us to understand our brain, to understand ourselves. And that's why I want to congratulate all of you, and especially the teams that are part of this country, especially Centro Nacional de Supercomputación de Barcelona, Universidad Autónoma de Madrid, Universidad Politécnica de Madrid, especialmente el doctor Javier de Felipe, la Universidad Rey Juan Carlos, Universidad de Granada, Universidad de Barcelona, Universidad Pompeu Fabra, el Consejo Superior de Investigaciones Científicas, la Universidad de Castilla-La Mancha y la colaboración del Instituto Cajal, del Consejo, Universidad Complutense de Madrid, IRB de Barcelona y el Instituto de Neurociencias de Alicante, que es mi tierra. Thank you so much to everyone. I congratulate you. Keep working and please give us the answers that we need to know. Thank you so much.
Se levanta la sesión. Muchas gracias.
Her Majesty, authorities, colleagues and friends, thank you very much for coming. It's an honor for us you come here just to support our research. Just, I would like to say a few words to introduce Idan Segev. Of course, many of us know him very well, but for those in the room that they don't, suffice to say that he's one of the most recognized, one of the most important computational and theoretical neuroscientists in the world, and is also one of the fathers of the Human Brain Project. His work is published in the top journals as Nature, Science, Proceeding of the National Academy of Science. Idan is the David and Inez Meyer Professor in Computational Neuroscience and head of the Department of Neurobiology and director of the Max Planck Center at the Hebrew University of Jerusalem, where he received his PhD in experimental and theoretical neurobiology back in 1992, many years ago. So today he's going to talk, introduce to Her Majesty and the authorities the Human Brain Project in 20 minutes or so. The title is Into the Future, the Human Brain Project. Dan, thank you very much.
Thank you very much, Javier. Thank you very much, all of you, for coming. And thank you very much, Her Majesty, for being with us. I actually met you many times in your museum. I was visiting there each time I was visiting Madrid, and your great artists, Pablo Picasso, Juan Miró, Salvador Dali. So I'm happy to meet you in person. And also, I read a lot about your institution here, the Alzheimer institution that you established, which is a wonderful institution. So thank you very much for helping us to help those who need help. So I have a very difficult job today. They gave me strict 20 minutes to explain the brain, the Human Brain Project. It's not easy. I worked on this the whole night, and I hope I will do it well. But before that, I want to thank Javier de Felipe, who I consider myself to be the intellectual grandson of Ramón y Cajal. I will show you later during my talk what Javier de Felipe contributed to our understanding of the human brain. So I want to start with a citation of a great Greek philosopher and writer. What a strange machine man is. You will fill him with bread, wine, fish, and radishes, and out comes pain, laughter, and dreams. I want to talk to you about the Human Brain Project dream. And I want to also apologize to those, many of you, that I can mention the work that they are doing in this wonderful project, of course, because I have strict 20 minutes. So I want to start with who started the Human Brain Project? How did it all start? And then I want to discuss a little bit about the novel vision of the Human Brain Project. What is the fundamental key vision of the Human Brain Project in a nutshell, very briefly? I want to explain something that is not easy to explain. Why do we need to simulate mathematically something that already exists? I have a piece of a brain here, and why do I need to put it in the computer? What will I learn from putting this into the computer? This is my big challenge in my general talks, to explain these things that is very, very clear to many of us, but very unclear to most of the people in the world today. And then I want to give you a few success examples, and eventually I want to end with the future of the Human Brain Project. So that's the plan for the 20 minutes. So how did it start? So the Human Brain Project actually started with this fellow. You heard a lot about him. Ingenious, complete genius, who realized something very fundamental that is not obvious at all, is that if you want to understand the system, you have to reconstruct it. In his case, it was with a microscope, very old microscope, and he reconstructed by painting cell by cell, branch by branch, in order to understand, while drawing, something about the structure. So he realized that reconstructing something that exists by painting it, in this case, gives an understanding, provides an understanding for the type of cells, for how they relate to each other, who may be connected or not to the one another, although he did not know that there are connections between cells at that time. And as I said, he was very poetic, but he realized this fundamental issue that while building, you understand. While composing things together, you understand the system you are trying to explain. And as I said, he was a very, very poetic. I always like to read his wonderful words. So he called the neurons, the different neurons in the brain, butterflies of the soul. It's so poetic and so romantic, and I guess that's the way to say it in Spanish, but I don't dare to try even to say it. But he realized something important. Cajal already himself, Ramón y Cajal already understood something very important. And this is a drawing by Ramón y Cajal, and you see these arrows, these red arrows that he drew 100 or more than 100 years ago, and these arrows mean that he misses something. He realized that there must be connections between the fibers, There is a fiber here that somehow should transmit something to the other one, and that there is a flow of some information, some kind of flow. And he had a whole theory about that, but this he could not measure because he was a pure anatomist. He didn't measure brain activity. He just drew the beauty of the brain, the butterfly of the soul. So he already then realized that we have to add activity and we have to add connectivity to his reconstructions. But he didn't have the tools for that. Now we do. So if Cajal would have sit here, he would absolutely be very, very joyful and enjoying what we are trying to do in the Human Brain Project. So we are going to extend Cajal's vision because now we have the tools to do the following. We can reconstruct and preserve, because we don't have the original data of Cajal anymore, we have only the drawings, but we don't have the data itself. But today with the computer, we can both reconstruct anatomically and also preserve brain data. When I say brain data, I mean not only anatomy, I mean also brain activity, I mean also brain connectivity. We can now use this digital world in order to preserve information forever. That's a huge, fantastic thing that we can do, that all the data in the world will be collected. That's one of the vision of the Human Brain Project, is to collect data in a platform that will enable future generations, everybody in the world, to approach this data, use this data, because it's always there. That's one vision. The other vision is to take this digital data and to model, to simulate brain activity. I want to claim that by simulating in the computer activity of the brain, we will understand diseases. We will understand the brain. We need to copy the brain into a computer and run the computer, and I will show you some examples, and this will lead to understanding. That's the second concept. And the third concept is that we can use the brain as a machine, as an example of a wonderful computer. Because I'm a computer that came from Reina Sofia Museum to give you a talk. I'm a computer built of microchips. So we can use this understanding of the brain to build future computers. So that's the third concept of the Human Brain Project. And all this came in the brain of Professor Henry Markram, who is sitting there, and he was really the one who took the seed of Ramon y Cajal and brought it to the 21st century. So, as I said, just to summarize, the Human Brain Project is built from future neuroscience, building atlases of the brain, digital information, future medicine, using this data in order to simulate the brain and understand how the brain is behaving in sick and in disease, and also the healthy brain, of course, and then build future computer inspired by the brain. This is a summary of the Human Brain Project in a nutshell. But the problem is that today we have to really connect many levels of data. We know about genes a lot, as we just heard from the minister. We have proteins, and there are connections, circuits, a whole regions, and so forth, until you have a behavior. So I'm behaving now, and Rudolf Nureyev is also behaving now, because you can see. So how is the brain of Rudolf Nureyev can produce this wonderful, amazing dance? How can we talk? How can we feel? How can we discuss things? Of course, I'm not going to talk about Rudolf Nureyev, but I'm going to talk today more about what we did succeed to do at the level of circuits. So here we decided, so this is the beginning of the Human Brain Project, the Blue Brain Project. We decided that this is an interesting piece of a brain. So in each one of our brain, below the skull, there is this a very particular circuit of cells. So in a 1 cubic millimeter size of a pinhead, just below the skull of each one of us, including the rat, including the mouse, including the monkey, there is this amazing circuit, very, very prototypical circuit, that includes about 30,000 cells in the rat per cubic millimeter, 40 million connections, synapses, and about 4 kilometers of wires. We said, let's start to show that we can simulate this thing in the computer and understand, or try to understand, how information flows and how many pieces of cells there are there, who is connected to who. We were trying to reconstruct this piece, This is the Blue Brain Project initiation by Henry Markham 10 years ago or so. And this is what we started to do. So first we started to do a modern Cajal work in order to characterize how many cell types. So this is a forest of cells, like Ramon y Cajal liked to call the brain forest of flowers, and this forest has many types of trees. Like you go to a forest and you see many types of trees. There are many types of cell types in the brain. We know today that there is about 55 different cell types in the brain, morphological cell types. But we next also need to record activity, because I said anatomy is not enough. We need also to see activity. And today there are new technologies. One of them is this intracellular recording of an electrode, of an activity of a single cell in the behaving brain. And this is how it sounds if you connect the electrical activity of this cell to a loudspeaker. It sounds like this. So this is what happens in each one of your brain cells. It's doing something music like this. So you see the brain is composed of electrical elements. Each one is doing tak, 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 tak. But there are many types, so many instruments, and there was a whole philharmonic symphony of activity, and this activity eventually makes me recognize you. This collective activity. So we need to put this collective activity into the model, not only the anatomy, but also the physiology. So that's what we are starting to say. So you can see that this cell likes to do ta 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 ta. This other cell is doing ta 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 ta. The other cell is a stutterer, ta 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 ta, stop, ta 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 ta, stop. So you see different types of electrical cells. There is very different type of morphological cells. We need to combine all of it together, and we do it via mathematical modeling. So Human Brain Project decided that theory and mathematics is extremely important. We cannot understand the brain without theory and mathematics, and this is an institute that the Human Brain Project built in Paris, and Alain Destex, somewhere here in the audience, is in charge of this institution in Paris, which is doing modeling. And in terms of, and doing modeling, you see, I can't trust my computer. Doing modeling means writing equations, and I'm myself a mathematician, writing equations to try to describe the electrical activity, because electrical activity can be described by electrical elements, by capacitance, by resistor, and so forth. So these are the equations that we are trying to write in order to replicate the activity. What do you want? So here is a success example. So I get from Henry Markram team in Lausanne through the internet a real activity of a real neuron. This is the brown spikes, we call them spikes. Tac, tac, tac. This is a particular cell type in the cortex of the rat. And then we build a mathematical model, and the result of the modeling is the green spikes. Tac, tac, tac. You see how similar we succeeded to model mathematically the electrical activity of that particular cell. And we succeeded to do it to all the cells in the cortex. So we now have the whole mathematical model of all the cells in the cortex, and now we can put it together in the computer. And that's exactly what we do with this supercomputing power. You take each cell separately and present it mathematically into a microchip. One cell in this microchip, another cell in this microchip. We started to combine all the information, both anatomical and electrical, into a model in the computer model. And then we get a circuit, connected circuit. And it looks like this, and here is a recent success. First of all, you see for the first time a voyage into the digital model of the brain. You are now taking a trip, a voyage, into a reconstructed digital reconstruction of the brain. You can see the jungle, that this jungle gives rise to our behavior. Quite amazing anatomical jungle. Now we can also do the simulation electrically. So now you see the activity, electrical activity. So red means a spike, blue means no spike. So you can see the symphony of activity. This network is really doing much more than a Mahler symphony, because it has 30,000 instruments, and these 30,000 instruments together generate this huge activity. And this huge activity could be a feeling of love, could be a feeling of appreciation of something, but could be sickness. Something may get wrong in this activity, and then you get some sickness, Alzheimer, Parkinson, epileptic seizures, sleep disorders, depression. So we need to understand through these simulations what goes wrong in the circuit when one gets sick. And we started to do it because we know that many of the diseases start, for example, epilepsy, start with a focus of activity that then spread like a wave. In the case of epileptic seizure, it's very obvious that there is a wave of spread of activity and the patient is epileptic. We want to understand epilepsia within our model. And when we understand epilepsy in the model, you can of course understand which are the parameters, which cell types, what connections goes wrong in order to generate this wrong activity in epileptic seizures. So this is one example. The next beginning, which is really heroic and very difficult, is to try to model not the rat brain, but the human brain. I spoke about the rat brain. There we have all the cells. But in the case of human, we are not going to penetrate the human brain and record activity of cells, of course. So what do? So there is a beginning due to the Human Brain Project, very much pushed by the Human Brain Project, to use material from operations that are being done in any hospital in the world. But most of the time, these pieces of the brain are not being used for science. Javier de Felipe, and actually Ramon y Cajal himself, was using human cells to try to understand, is the human brain cell similar or not similar to the rat brain? Is there something unique in the human brain? So we have in operations, you can see a big piece of brain was taken in operation, such a big piece of a brain, because there was a big tumor, and you can compare it to the brain of the rat or a mouse. You see that the piece that they take from the operation is actually bigger than the whole brain of the mouse. We can use it, and we are starting to use it as a tissue to understand the human brain. You can see people in Amsterdam that we are working with. They are part of this group. They are trying to reconstruct the anatomical structure of human cells. You can see that also they have different type of cells in our brain, like in the mouse, but many other variability of cells. And we are using, of course, without Javier de Felipe, we cannot even move, because Javier de Felipe, as I said before, he is really the grandson of Ramon y Cajal, and he likes to look at the very, very specific structure of human cells. This is a human cell. This is a human pyramidal cell from the cortex of human, from temporal cortex of human, and you can see that he reconstructed in such high resolution, and you see really actually these little things that are called dendritic spine, and that's where the connections, the synapses between one cell to the other are formed. So you can see a little piece of a brain from Javier de Felipe, from a patient here in Madrid, taken out, and you can really reconstruct now the whole neuron. And we have many neurons, so we are starting to build a model of a human brain circuit, not only of the rat. It's a huge jump, and I'm very pleased to be part of it myself. We are also using information, or starting to using information coming from Germany, from Katrin Amutz, because she recently developed a new technology to trace connections between regions. So one region of my brain and another region of my brain is connected by fibers, but we didn't have the resolution to see individual fibers connecting one region to another. This is a very recent paper coming from Jülich, Germany, showing the connections between regions, because eventually if we want to build the whole brain, of course, we need to connect regions, and now we have a beginning of ways to connect the brain. And eventually, and I want to end by that in a second, I want to emphasize again that we want to understand all these diseases, terrible diseases. So our brain is wonderful, but very, very fragile, as you know well. Each one of us know well how fragile it is. Sometimes I wonder myself, how does it even function with all this fragility? Seriously, with all these complexities, how does it function? So we really need to understand what happens in each one of those 560 neurological diseases that we know what goes wrong in this circuit. And I think that without reconstructing the circuit, step by step in order to understand its structure, the elements it is built from, the building blocks, and the activity, we are not going to understand diseases to the depth. But I want also to say something, that we need to talk to the society, and you need to educate people. Some people are afraid of big projects. They think that we are going to penetrate the brain or things of that sort, and they are right to be worried because we need to explain what we do. And so the Human Brain Project came with a wonderful idea, and we just met recently a representative of the 3,500 museums, science museums in the world. These people are interested in presenting our results to the public. We shall devise a brain corner in each one of these museums. There will be a corner, a digital corner, where we can send our data digitally to all over the world, and each country will take this data, will transform it to its own language and the way they like to present it, but we shall be the supporter of the brain corner for families, for children, for youngsters to understand the brain. So that's another direction, and this is being taken by Maya Levy in the Jerusalem Museum in Israel. And of course, eventually, we want to build a platform, and we have Sean Hill here, who is working very hard on this informatics to put all the data in a way that everybody in the world can access this data and make use of this data for his or her own research. So this platform should serve society. Like we have the genome today, everybody can go and see the genome that was sequenced. We want to have the brain data available to the public. That's another big vision of the Human Brain Project. I want to thank you very much. I think we are going into a wonderful, near, amazing future. It's waiting for us. I think the main thing is that we shall be able to learn to collaborate. Muchas gracias.